lock the door.
Don't you have to ask me if
Just lock it
you
up.
want
It's
to?
fine.
No, that's a university. We call it university. That's not a high school to ask the teacher for going to the
If you
washroom.
let me, I'm gonna go to the toilet.
I will. Okay. So what type of B cells exist in our body? First of all, we can distinguish B cells in two types, B1 cells and B2 cells. You heard about B1 cells because you have to learn about the natural antibodies. Do you know what is natural antibodies? It's again PAMs. So these are not the member of the innate immunity, rather adaptive. So not adaptive immunity, the innate immunity. So B1 cells produce the nature antibodies, and we can distinguish also the B2 cells. One population of B2 cells called marginal zone B cells, which localized to the which tissue? No.
What?
Yeah, what? It's not exactly a lymph node. Okay, and also the conventional B cell is the member of the B2 population of B cells. Okay, what are the main characteristics for B1 cells? So they are the main cell type which produce the natural antibodies which have the isotype of IgM. This isotype exists in our body, so produced by plasma cells when they not in touch with T cells, so in T-independent cell activation. Because the isotype switching need link of T cells, so need the help of T helper cells. Okay, so they are longer life plasma cells, and they can also renew. Okay, B2 marginal zone T cells, they located in the spleen, as you told, and it's very important that they activated by polysaccharide antigens, and yeah, but it's more important, they also long life population. Okay, the conventional B cells are the most important population of B cells, and they can activate it in T-independent and T-dependent way. It means there is two ways of T-independent activation. First of all, you told that on the surface of B cells express BCR and also PRRs and also complement receptors which help in the signaling through the BCR. So when this B cell recognizes an antigen, not only the B cell PCR receptor will activate, also the complement receptor and also PRRs will activate and enhance the activation through the BCR. Another way of activation of T-independent activation is when more BCR on the B cell surface activated in the same time. So they cross-linking the antigen, and we will get the enhanced signaling in the cell. So this is the two type of the T-independent activation. What are the main characteristics for the T-independent B cell activation? So they will produce, the plasma cells will produce IgM isotype because they didn't match with T cells. Therefore, they will have low affinity because they don't have affinity maturation, and the effector plasma cells will not have memory because of they didn't match T cell. but it will be a rapid response against the antigen. And it's very important that this activation occurred out of the follicles. So extrafollicular activation is the type independent, T cell independent activation. Okay, so B cells are antigen presenting cells. Where are the antigens? In the follicle, there are the antigens are in immune complexes. You well know that in immune complexes there are antigens, immunoglobulins, and also complement fragments. So these three members exist in the immune complex, and FDCs, these are the follicular dendritic cells, bind these immune complexes on their surface. When they recognize the antibody in the immune complex, they bind to the Fc receptors. When they recognize the complement fragment in the immune complex, they bind with their complement receptors. Okay, and that is the function of these FDCs. So they present the antigen to the B cells, but also contain these immune complexes for neurons. Yes, these are the receptors on the FDCs. When these cells with enough affinity can bind this complement, so this immune complex, they will get from the surface of FDCs this immune complex and will activate it, but it's pre-activation because they need also T cell activation. In the edge of the follicle, in the primary follicle, first B cells is activated by antigen, which localize on the surface of FDCs, and also T helper cells are activated by the dendritic cells, which enter the lymph node and came from the peripheral tissues. So these two types of lymphocytes are reactivated but need another signal to the maturation, so to the proliferation and differentiation.
The activation of both has to be done with the same antigen,
yes.
right?
Yes, it's very important. So these T cells and B cells are activated by both, by the same antigen. Okay, CD40 is the most important costimulatory receptors. Their ligand called CD40 ligand. The ligand is on the surface of T cells, and the CD40 receptor on the antigen presenting cell, in this case on the B cells. And the activation through this costimulatory receptor lead to the proliferation of B cells and also the T cells. Okay, so first the B cells are activated through the immune complex localized on the surface of FDCs. T cells activated through the TCR and through the MHC2 peptide complex on the surface of BCs, and they met on the edge of follicle. And when the B cell receptor affinity is high, they can differentiate and proliferate and differentiate to plasma cells extrafollicular. But when the affinity is lower, they enter the follicle and start the proliferation and differentiation intrafollicular. So the affinity is the main thing which decides which way it will activate. Yes, so they recognize the same antigen. Okay. What happened in the follicle when B cell intact after the activation by T cells. Yes, we were talking about this. I think you have to know these receptors, adhesion molecules. ICAM and VCAM is on the surface of the antigen presenting cell, and the component adhesion molecule LFA and BLA on the surface of FDC. Yes, this is the immune complex which reactivate the B cell. Yes, B cell remove this immune complex from the surface of feces and will present to the T cell. But it's a competitive mechanism because, again, the affinity of BCR are responsible for the T cell presentation. So BCR will activate through the immune complex, and in the cells binds to the MHC2 class 2 of MHC, and will present it on the surface in a complex, so antigen and MHC2 complex will recognized by the T helper cells. Yes, through the TCR. And this is the first signal for the T cells. And T cells will produce in response to the activation, the first step of activation. So they will produce different cytokines which enhance the proliferation and also the isotype switching, for example, of B cells. Yes, these are the most important co-stimulatory molecules between the B and T cells. Okay, I don't know, you don't have to know this, I think, Islam and the ICOS. Okay. So the first step is the activation by the antigen. After that, the first signal generating the T cell, the production of different cytokines which induce the proliferation of B cells. And B cells will differentiate to plasma cells, which will produce different type of antibody, but against the same antigen. And also they can differentiate it to memory cells. And what is between the isotype switching and the maturation also need to the differentiation, so the effector function for the generation, for the developing of effector function of B cells. So what is the affinity maturation? The variable site of immunoglobulin have different fragment in the gene called CR1, 2, and 3, but you don't have to know. And in this fragment of gene can be mutated the basic pair, okay? And it will be it will lead into the affinity maturation, which means not the different specificity. So the specificity is the same, but the affinity is higher after the conjugation. So this is the follicule, so secondary follicule. Because in primary follicule, B cells are in, so B cells not proliferated. So this is the secondary follicule. The dark zone is the place of the proliferation, differentiation of B cells. The light zone is the place of the antigen presentation and the competition for T helper cell. after the activation, B cell can differentiate to memory B cells or plasma cells. Plasma cells will live for years and produce the same antibody against the same antigen. Memory cells also long-life cells and can be activated when the specific antigen enters the body. And the B cells which have lower affinity to the same antigen, they will back to the cycle and go through again the maturation, so the affinity maturation. So in germinal centers occur the tunnel proliferation. Yes, I told you that not only the B cell, also the T cell will activate and proliferate and differentiate. The somatic mutation, the affinity maturation, and also the isotype switching. What is the isotype switching and what it's so important for the B cell populations? Okay, it's another slide. Also, autoantigen can be entered to, yes, so this is a lymph node, and also this is a lymph node. And when PCR specific to self-antigen, it will react by autoantigen, but normally in the lymph node, the specific antigen is not exist. So the costimulatory signal through the T cells won't be developed, and therefore these B cells will die. But in the case when we will get from the T cells and also from the FTCs, the second signal for the proliferation, these B cells start to develop and differentiate, and autoimmune diseases will develop in our body. Because these plasma cells will produce antibody against our antigen. Okay, isotype switching. So you know that immunoglobulins contain light chain and heavy chain, and both contain also variable and constant region. Variable regions also have effector function, so they bind the antigen and form immune complexes. But the complement, the constant region also have effector function. They bind to the Fc receptor. And the isotype determines the specific Fc receptor which binds this antibody. So the constant region is very important in the isotype switching. You see that there are 9 fragments of constant region, and different combination of this region determine the isotype of the producing antibody. Okay, Fc receptors. Different cells express various Fc receptors. For example, mast cells express Fc epsilon receptor, which recognize the IgE isotype. For example, macrophages express Fc gamma receptor, which recognize IgG, that you well know this. Okay, and before birth, also baby has antibody in their body. It's mother antibody, isotype of IgG. After birth, through the milk, baby will get IgA isotype of antibodies, and developing the IgN and IgG producing plasma cells in the baby after 1 year and later. the IgG is the first on the surface of B cells, and the plasma cells produce IgM without the interaction of T helper cells. We well know that in parasites infection and also in allergic reaction, IgE will produce, and IgA play important role in mucosal surface protection. Okay, and this is the last slide. So the first step in the activation of B cells is the antigen binding through the VCR receptor, and they are present in a complex, this antigen to the T helper cells. In this case, because it happens, it occurs in the intrafollicular, these T cells called follicular T helper cells. And this is the first signal for the T helper cells. It will produce IL-2 and IL-4. IL-2 is very important in the self-activation, in the life of T cells, the need to proliferation, but also start to produce IL-4 in this case, and will activate the B cells through the IL-4 receptors, and this is the second signal for the B cells, and start to express also B7 on the surface. So CD40 is constitutive on the surface of B cells, but CD40 ligand on the in the membrane of T cells will enhance via the first signal, and this co-stimulation helps to B cells enhance the expression on their surface, this co-stimulatory molecule, and the complement of this molecule is the CD28, which also again signal for T cells produce this cytokines. So it's a complicated mechanism. But finally, when B cells are activated with IL-4, they will produce the isotype of IgG. When they activated with interferon gamma, they will produce IgG. So it's very important that T cells determine the isotype switching of